Hi, it's me, Hannah, again, and this week's module's contents is all about COVID-19. And in this video, I'll be focusing on the Rt-Pcr tests. So let's get started. COVID-19 is a disease caused by SARS-CoV-2 and was discovered in 2019 in Wuhan, China. It is still very contagious and has spread rapidly around the world. Although COVID-19 primarily affects the respiratory system, much like a bad cold, flu, or pneumonia, it can also affect other parts of your body, not just the respiratory system. Most people infected have mild symptoms, although older adults and people that have underlying medical issues are vulnerable to severe illness. Hundreds of thousands have died just in the United States. One of the most important aspects of our battle with COVID-19 is not just the development of the vaccine, but the development of the Rt-Pcr technology. This process is instrumental in determining individuals that have COVID so that protocols can be put in place to prevent the spread of the virus and treatment of the infected individual. The nasal swab test is the very first step in isolating the coronavirus. The sample, which is taken from the nasal pharyngeal region of the individual, is examined. Because COVID uses messenger RNA as its genetic material, a traditional PCR that detects pathogen like bacteria or other viruses that use DNA in their genome cannot be used initially. The first step in the Rt-Pcr is the reverse transcription of COVID-19 RNA to DNA. This is accomplished by adding several chemical solutions that remove proteins and fats and that only extract RNA, which contains your RNA fragments and the viruses, if present. Reverse polymerase is used to make complement DNA strands from the mRNA strand. Now that the viral RNA is now complement DNA, a traditional PCR portion of the test is performed. Scientists do this by adding short fragments of DNA called primers that are complementary to unique ends of the unique genes of the viral genome that are used as identifiers for the virus. These genes include one that is responsible for a protein that protects the viral genome, and this is called a nucleocapsid protein. If the virus is present in the sample, these fragments attach to the targeted viral DNA sections. This DNA is amplified during the PCR process. This process amplifies only one segment of the targeted viral DNA. The process of denaturation, annealing, and extension is repeated 30 to 40 times, exponentially increasing the viral DNA segment and hence its detectability. For example, a chemical that is added would fluoresce when exposed to UV light if there is COVID DNA. That would be considered a positive test. If no fluorescing occurred, the test is negative. This means you probably didn't have an infection when tested. However, a PCR test can still be negative if you contracted the virus with a short time before being tested or even after a week of having the infection. This test, although sensitive, works best during active infections, not past ones. That concludes my answer to my friend's question, and the sources that I used to compose this talk is from the CDC, Center for Disease Control, their articles on vaccines, and the International Atomic Energy Agency, Rt-Pcr articles, and I utilized all of the videos from the module this week. And thanks so much for listening, and have a good one.